Good afternoon, everybody. I think we are having another very exciting panel. So we talked this morning about the NF2 actionable targets, and now it is up to an expert pain panel to talk about schwannomatosis and in actionable targets in pain in schwannomatosis. So Larry, I'm gonna hand it over to you to moderate this very exciting panel.
Great, thank you, Annette. So I think what I'd like to start with, since we do have limited time, is some of the big questions that the panelists and I have discussed earlier. And one of the questions, I think, is really trying to get a better understanding that's going to help basic scientists understand where to go next regarding the real nature of this pain. And I guess maybe we could throw it out to Laura or somebody else just to talk about your impressions of that. And I know Scott Plotkin's here too, and he might, and some other clinicians on the line as well, who might have some insights into that. But can we start with my take on schwannomatosis pain is that we still have a lot to learn at the level of natural history and the types of pain and really, again, segregating who gets pain when and where and how. Anybody else want to take a stab at that?
Well, so of course that's been the biggest problem is why are some tumors painful and others are not? And I think Sheila has some data, or we've published some data with Sheila saying that there are some location relevance to what are more painful than others. But in the periphery, it's hard to say what makes a tumor painful and what makes a tumor not painful. Because I think there were some questions between spinal cord schwannomas being not as painful and things in the periphery being more painful. Is that correct, Sheila?
That is correct. Definitely tumors that we profiled from the periphery, specifically the legs, were apparently more painful. Statistically significant number of those tumors were painful, but I guess we can all agree that collection of tumors by surgery, it also depends on whether the patients report presence of pain, and that's why the surgical intervention is performed. So it's really hard to tell, I think, although we found a correlation that these tumors were indeed biologically different and that's why they were more painful, or is it because of accessibility that they could be surgically removed and they were more painful and that's why they were collected. But in addition to tumor location, we also found significantly higher proportion of female individuals reported pain. And also we found some molecular differences in tumors that were considered more painful from schwannomatosis patients compared to non-painful tumors. So we found like several signaling pathways were upregulated in these tumors, and we also found that although these tumors didn't have a high mutation burden, they had higher copy number alterations at the genomic level. So there's certainly molecular differences in addition to phenotypic differences in these tumors, and they require further exploration.
I think that's really interesting because there's been data showing, for example, TLR4, toll-like receptor 4 mediated pain. There are sex differences, big sex differences with regards to how inflammatory mediators, for example, influence pain in men versus women or males versus females in general. And so your molecular studies, I think, are going to give us some insight into the degree to which that's common. One question I had is, does anybody on the panel have an experience understanding not just schwannomatosis tumors, but tumors in general? Are tumors in general more painful in the legs? Has anybody, I can't recall seeing anything about that, but.
No, not that I know of.
Yeah, I don't either. So this that's another question that we may want to put on the list is this unique to schwannomatosis or other? There aren't a lot of syndromes where you have a lot of peripheral nerve tumors that are purely schwannomas, of course. So then I'm just wondering if there are examples where people have said that a painful tumor, which dermatome is most painful when you have a tumor growing on a nerve or a tumor in general.
Right.
I think the other big issues that we were talking about, coming back to the molecular profiling, was both regards to tumor-associated pain, but also the pain that's not necessarily tumor-associated in schwannomatosis. what is the underlying cause of that. And our lab, and I know at Match, our lab, some of the data from our lab and from Kim's lab certainly are matching in that regard, and other labs as well, I know are looking at this too, suggesting that the mutations in the Schwann cells may be reflecting things like recruitment of immune cells that then could subsequently influence pain through chemokines and cytokines, or the Schwann cells themselves may be in doing that. And maybe, Kim, if you want to summarize some of your data in that regard.
Yeah, actually, I'm going to share my screen, if I can. Here we go. So can everybody see the slides that I put up?
Yeah.
Okay, so what we've been doing, and Larry has been doing similar things in mice, and we've been doing them in humans, is once we have a surgical case where they extract the tumor, we have been to kind of going along with what was mentioned this morning about looking at mutations and kind of classifying schwannomatosis into different subtypes based on what their mutations are. So we've been looking at SMARCB1 and LZTR1 mutations in these patients. And then we've been taking the cells from the tumor, growing them in the dish, and then taking off the conditioned media just to see what are these cells, what are these tumors secreting? And do they secrete something that acts on sensory neurons that makes these neurons fire either spontaneously or make them more susceptible to painful stimuli? So the one thing that we've been wondering about, let me go to my next slide, is that so we found all these cytokines, and Larry can can go into also some detail on this. And I know Sheila has found similar targets in terms of inflammatory substances. So what we've seen is that there are some differences between the in painful schwannomas with the different backgrounds, genetic backgrounds. So we've been focused so much on what makes these different, but we also really, when it comes to drug targeting, I think we have to start looking at what makes them similar. And a lot of that is that the fact that a lot of the cytokines act on the same downstream receptors in the neurons. And we were focusing on then looking at what are the secreted proteins, and what do they have, what downstream targets and receptors on neurons that are affected by these. And we focused a lot on TRPV1 and TRPA1, and the pathway being that cytokines will either act directly on TRPV1, TRPA1, or something would, and looking at downstream calcium signaling and seeing if those make the pain more pronounced. So, but going back to the earlier session today, which Sylvia was talking about NF-kappa B. Well, this really inspired me because this was something that's been on my mind and thinking about, because NF-kappa B not only regulates the genes in the schwannoma that are being secreted, but also on the receptors that we have been finding in the downstream targets in the sensory neurons. So I think that might be, and I'm not a person who should by any means suggest clinical trials, but I think on the basic science side of it is something that maybe one of the drugs, the ADASA, that was suggested as a NF-kappa B inhibitor, and something that has been used and is FDA approved, might be something to look at on the basic science side going forward, just because it kind of brings it all together into one node, which is something that, when we're looking at, how many patients can this help, would be a remarkable thing. So if anyone has any other suggestions, I mean, we've been throwing targets out there and trying to figure out, should we look at this target, that target, how specific should we be based on mutation, and it's just getting to something that is a good consensus. It would be very valuable. I don't know if anyone else has anything to say about
I can complement your data there, Kim, and say that our mice that have Schwann cell targeted loss of SMARCB1, and then our mouse cells that we recently I just started culturing that are also LZTR1 null, we have similar profiles, and we've been able to see similar things in schwannoma cells from patients that we got from Cristina Fernandez-Valle. And so I think our data are lining up to some degree. And I think you're right. I think there are maybe some common targets. It'd be interesting to know to what degree we could actually see changes in pain responses in the mouse models. That would be there. There's a question from Marco. What should a natural history study on pain and schwannomatosis look like? Does the scientific community need to agree widely on that? I would argue that a natural history study should probably give us some great insight into, again, the variety of pain. I think we've got a good start. There's the paper that Scott Plotkin was involved with, but I think we need more data along those lines to understand the varieties of pain, to what degree we're talking about tumor-associated. Is the tumor-associated pain really different from the pain that's not tumor-associated? Those are kind of the questions I'd like to know. Kim and Sheila and Laura, any ideas along those lines?
Well, no, I think that something that we still miss is to have a, frankly, genotype-phenotype association also. There are preliminary data that LZTR1 schwannomatosis might be more painful associated than the other one. But however, in my experience, there are some patients that have mostly painful schwannomas and others that are no painful schwannomas at all. However, what I think that it might be very difficult to define this genotype-phenotype correlation is the number of patients that would be needed to have a statistical significant association of, SNP polymorphism. We don't really know what we can find.
That's a good point, and with a rare disease, that's always hard to do. Having said that, Scott Plotkin just sent a comment that it sounds like they've got greater than 5 years of data along these lines, and working on getting that out. So I'm hoping that can help us all. Also, a question from Scott, by the way, Kim, for you, asking if you've got data in animals.
So my data in animals, we've actually been just starting to work on this. As we're taking the media that was the factors, in the media that have been secreted by the schwannoma cells in culture, and we've taken media from painful and non-painful tumors. We've injected them into the foot pad of mice, and the mice that had received the painful condition media spend more time licking their paw, shaking the paw, and more into kind of an acute pain. Then we've also been looking at whether there is evoked pain and increased evoked pain in these animals. And we see statistically significant differences in mice that have received painful media versus a non-painful in mechanical force. They have much more sensitivity to light touch, and that is something that we've been looking at. Now, we've also started looking at thermal sensitivity, and we're not seeing as much of a difference, but our ends are kind of low on that end. So that's ongoing right now, and something that we're just kind of moving over to the animals to look at. And that's really been our only way of assessing the pain in animals.
So, and our animal models, which are both the knockouts, we're consistently seeing very high sensitivity to capsaicin in both knockouts. So we just started looking at the LZTR1 null animals. We've already done the cell work there, but both animals. But what's interesting is it seems like you've got differences with regards to the mediators that are coming in both models. So we're trying to tease that out a little bit. but they do both have the pain response. So we do see thermal sensitivity, but kind of like what you're saying, Kim, it's variable, and it seems like for us it depends on when you knock it out. So the longer they've been knocked out, the more recovery we get from that. And that might be Schwann cells that are turning over, but it also just might be tolerance that they develop over time. So it's a complicated thing. Annette asked a question for Laura. How many patients do you think you would need for a phenotype-genotype correlation study?
Well, it depends what we are looking for. If we are looking for just a correlation with SMARCB1, LZTR1, or NOS, schwannomatosis not associated with these two genes, maybe a few hundreds will be enough. But if we are looking for polymorphisms that can be shared in painful and non-painful patients, I think we need a few thousand of patients.
Yeah. Annette also was wondering, would it make sense to do tissue microarrays or RNA-seq types of studies from tumors, kind of along the lines of what Kim has started doing, I think. I guess a bigger question for me is, I don't know how possible this is, but do we have ever times when we get non-tumor associated nerve, even adjacent to the tumors, that could be examined in that regard for differences? Or is it always going to be, since they're encapsulated, I imagine that's not something you see?
Right. No, it's all like most of the surgical specimens are encapsulated tumors, and then they're really trying to spare the nerves, so not to cause neurological deficits. So to see that interaction type thing would be might be difficult in humans. It might be different in mice if the mice are developing tumors.
And Scott just raised a really great question regarding possible biomarkers that we could be looking for in a clinical trial population. So could we use blood, for example, or CSF or saliva? I guess that's an interesting question, is because if really even the hemizygous Schwann cells that are not in tumors are secreting proteins that contribute to pain phenotypes, those could be detectable in serum, for example. And so that would be an interesting thing to know. But of course, the other possibility would be to do something a little more complicated, which would be kind of along the lines of growing the tumors. Yeah, that's so cytokine levels, exactly, Scott. That's what we're thinking, since we're seeing these cytokine profiles changing in these painful tumors. So I think we've addressed some really interesting questions here, both at the basic science and the clinical science level. And I would argue that the actionable items are to continue talking between the clinicians and the basic scientists going forward about the best ways to approach a possible biomarker study, as well as the natural history of pain. But although maybe Scott's got that in the new paper that he's writing right now, and then for all of us to talk about some of these markers that we're trying to find and see if it makes sense, and if we could use a combination of some of these models to screen for drugs that can reverse these phenotypes. And it may be a combinatorial therapy approach that we might be looking for, given that there are a lot of things changing in these Schwann cells. SMARCB1 in particular, we've been able to show it's directly regulating the transcription of things like IL-6 and IL-1 beta and everything else. And so that may be more than one factor we'd have to address. Well, I think we are out of time. That was a quick session, but I thought pretty informative. I want to thank everybody on the panel, and I also want to thank everybody listening in. And let's keep this discussion going, please.
Absolutely.
Thank you very much.
Thank you.